Come on and cry me a river, cry me a river. I cried a river over
Hello. How's everybody doing? I hope very well. Welcome to today's emission, live emission. Again, after a long time. Here I am again with some articles, always related to neuroscience, psychology. That's my interest. So, today I have one very interesting title to share with you. Let's start. Rotating brainwaves help the mind refocus after distraction. Rotating brainwaves, that sounds very cool. Let's see. As surely as the brain is prone to distraction, it can also return its focus to the task at hand. A new study in animals by scientists at the Picower Institute of Learning and Memory of MIT shows how that seems to happen. Coordinated neural activity in the form of a rotating brain wave puts thoughts back on track. The rotating waves act like herders that steer the cortex back to the correct computational path, said study senior author Earl K. Miller, Picower professor in the Picower Institute at MIT's Department of Brain and Cognitive Science. Okay, too many Picower in the article. Picower Institute postdoc Tamal Batabial, wow, where's that name from? Is the lead author of the study published November 3 in the Journal of Cognitive Neuroscience. Mathematical rotations. In the study, animals were given a visual working memory task, but sometimes they experience one of two different kinds of distraction while they tried to remember an object they saw. As one would expect, the distractions affected the animal's performance on the task, sometimes causing them to make mistakes or at least slowing down their reaction time when the task called on them to act. The researchers, meanwhile, kept tabs on the electrical activity of a sampling of hundreds of neurons in the prefrontal cortex, a brain region responsible for higher level cognition, to analyze how the neural activity varied as the animals performed the task over hundreds of sessions, with or without either of the distractions, and in cases where the animals performed well or not so well, the researchers employed a mathematical measure and visualization that measures their degree of coordination over time called subspace coding. Subspace coding shows that the activity of cortical neurons is highly coordinated. I didn't know that. I hope we can understand a little bit more about this in this article. Like starlings murmuring in the sky, Miller said. After the distraction, there was a rotation movement in subspace, as if the birds were circling back together after a disruption of their information. In other words, Miller said, the circling seemed to represent the recovery of their activity state from the distraction. The circling, this rotation, waves, I assume. Indeed, the rotations predicted the animal's performance on the task. In case where the distractions didn't cause a mistake, the neural data showed a complete cycle, indicating the recovery was complete. I would love to see an animation of this. Maybe we can try to find one in YouTube. In cases where the distractions caused the animals to err, the trajectory fell short of making a full cycle by an average of 30 degrees. The trajectory during errant sessions showed a slower speed, which could explain the lack of recovery from distraction. I'm starting to visualize in my mind. But we can try to find an animation. A related finding was that the animals were better at recovering if the time between the distraction and needing to act was longer. That makes sense. The brain needed that time. The data showed to come full cycle mathematically and get back on track behaviorally. The subspace coding data suggested that neurons work in a highly coordinated way and that this rotational organization helps maintain their focus. Notably, the rotations only occurred if there was a distraction. Both styles triggered them that the animals were trying to ignore. I couldn't read that very well. I'm going to try that again. Notably, the rotations only occurred if there was a distraction that the animals were trying to ignore. The rotations did not occur spontaneously. Reflect physical rotations. I have to find this, search this on YouTube right now. Let's see if we can have a notion of what that is. Oh, there we are live. Effect of rotations of mirror. Physics of microstations. Reflect physical rotation. Okay. Let's go back. Subspace coding is just an abstract mathematical representation of neural activity over time. But when the researchers looked at the direct physical measurements of neural activity, they found that it actually reflected a real traveling wave rotation across the cortex. Once again, mathematically speaking, through time. Multiple measurements showed that neural spiking activity had a spatial order with continuously changing angles. Damn, that's so hard to visualize or imagine. Consistent with a wave of activity rotation across the cortical electrode. In fact, the actual wave rotated with the same speed as the mathematically represented one in subspace coding. What was subspace coding? Let's ask Google. What is subspace coding? It's a method of error correction in digital communication, especially in networks coding. where information is transmitted as a set of vector subspaces instead of individual symbols. Okay, almost the same. I'm still not seeing it. Instead of protecting each data point, it protects the entire shape or subspace. Encoding, transmission, decoding. Okay, this is a very technological term, it seems, to me at least. Okay. Let's assume, or let's make us we understand perfectly what's happening here. Maybe we can understand some things, in the end at least. There's no reason in principle why a rotation in this mathematical subspace should correspond directly to a rotation on the surface of the cortex, Miller said. But it does. That suggests to me that the brain is using these traveling waves to actually do computation, analog computation. Analog computation is way more energy efficient than digital, and biology favors energy efficient solutions. It's a different and more natural way to think about neural computation. In addition to Miller and Batavial, the paper's other authors are Scott Brinkat, Jacob Donoghue, Michael Lundqvist, and Meredith Manke. Okay, that was all in this article. I have another one. Hungry brains rewrite the senses. Cheese and chocolate might not tempt a fruit fly's palate, but a hungry fly short on nutrients their smell carries a hidden signal. When deprived of certain amino acids, the building of blocks of protein, these tiny insects develop a surprisingly refined sense of smell that helps them track down not just food, but specific bacteria living in fermented foods. For years, scientists have known that the animals can sense when they are missing certain nutrients and will actively seek out foods to compensate. What was remained unclear is how the brain translates this internal nutritional need into a behavioral drive, how a physiological need becomes a sensory want. Like a man lost in the wild, when he has nothing to eat, he's gonna start to crave some insects because the proteins are there and his body knows it. This was the question that motivated researchers in CF's Behavior and Metabolism Lab, led by principal investigators Carlos Ribeiro, the study's senior author. A Tale of Two Genes. Sydney Sweeney genes. Animals, including humans, cannot produce all the amino acids they need. These so-called essential amino acids, or EAAs, must come from food. If even one is missing, the body's internal chemistry changes dramatically. Protein synthesis falters, metabolism slows, and the brain drives a specific appetite for protein-rich foods. In fruit flies, the behavior is easy to observe. Remove a single essential amino acid from their diet, and they begin to seek out yeast, their primary source of protein. But Ribeiro's team wanted to know what was happening in the brain to make this shift occur. Using synthetic diets that each lacked one of the 10 essential amino acids, the researchers sequenced RNA, ribonucleic acid, from the heads of flies in 11 different conditions, 10 amino acids deprived diets, 10 amino acid deprived diets plus one fully balanced control. This allowed them to track how the expression of thousands of genes changed depending on which amino acid was missing. Very interesting. Once again, why we cannot have visuals in these fantastic articles? Maybe we can stimulate our imagination in a better way, in this manner. It's a good exercise for our minds to try to imagine and visualize what we are reading. That is so hard to understand, to me at least. Let me know. Where were we? Okay. While the fly's behavior was similar across all amino acid deprivations, showing an increased drive to feed, each deprivation had its own unique fingerprint in terms of gene expression, says Gilles Ranivo, the study's first author. But despite those differences, some genes change in the same way no matter which amino acid was missing, of course. Among those shared changes, one pattern stood out. 2 olfactory receptor genes, both involved in smell, were consistently upregulated in response to amino acid deprivation. These genes, OR92A and IR76A, became the key to understanding how the fly's sense of smell is fine-tuned to meet their needs. Yeah, that makes a lot of sense. The yeast connection, how smell shapes flavor. The first receptor, OR92A, was already known to respond to diacetyl, a molecule that gives butter-flavored popcorn its distinctive aroma and contributes to the smell of wine and beer. Yeah, once I saw a documentary that talked about where's yeast in our food. It's everywhere. Yeast produces diacetyl during fermentation, and since yeast contains all essential amino acids, it makes sense that this smell would be especially attractive when flies are protein deprived. When the researchers test flies lacking OR92A, the insects could still locate yeast but fed on it less. They could smell where it was, but it didn't taste as good to them, as Ranivo explained. That's because olfaction isn't just about finding food. It also contributes to flavor and the evolution of palatability. The team went a step further, using a mutant strain of yeast that didn't produce diacetyl. The result was the same. Flies were less likely to feed. Losing that key olfactory cue disrupted their sense of flavor, making the food less appealing. Just as humans lose appetite or enjoyment of food when they have a blocked nose, flies rely on a close interplay between smell and taste to regulate feeding. Chocolate neurons in the brain? The second receptor, IR76A, proved even more mysterious. Searching through microbiology and food science literature, the researchers discovered that fermented foods such as cheese and chocolate emit a compound called PEA, which activates IR76A. Testing showed that flies' olfactory neurons responded strongly to the odor of chocolate, but not to cheese. Had they found chocolate neurons in the brain? Not quite. Since flies don't normally feed on chocolate or cheese, I strongly disagree. The team dug deeper and found their real connection. Both foods are fermented by Lactobacillus and Acetobacter bacteria, which also produce the same compound. That's when things clicked, says Silvia Henriquez. How many names in this article too? The study's co-first author. The flies weren't being attracted to the chocolate itself, they were responding to the bacteria growing on those foods. And those bacteria are also natural residents of the fly microbiome. Okay, now that makes sense. When the researchers exposed flies to live Lactobacillus and Acetobacter bacteria, the IR76A neurons responded even more strongly. Behavioral experiments confirmed the link. Amino acid-deprived flies actively increased their feeding on bacteria, but only when the bacteria were alive and metabolically active. Dead bacteria no longer triggered feeding. In other words, the flies were seeking the bacteria for their metabolic benefits. And when the team knocked out the IR76A receptor, the flies lost interest altogether, even when nutrient deprived. Wow. This was the most surprising finding, says Ezra Nevo. It showed that the fly's sense of smell was literally tuned to detect bacteria, and that this tuning depended on their internal nutritional state. Amino acids deprivation didn't just change neural activity, it changed which receptors were being made in the first place. Microbial allies in flies. Microbial allies in flies and in us. Why would a fly seek out bacteria when amino acids are scarce? Okay, I don't get the question. Let's do it again, please. Why would a fly seek out bacteria when amino acids are scarce? Okay. Previous research had shown that feeding on certain bacteria improves egg production in amino acid-deprived flies. Other studies have revealed that the gut microbiome can boost amino acid absorption under nutrient-poor conditions by producing enzymes that break down proteins more efficiently. By following their nose towards bacteria, it seems the flies have evolved to use microbes as allies, seeking out partners that increase their chances of survival when challenged by amino acid deprivation, says Henriquez. The parallels to humans are intriguing. Many traditional diets include fermented foods, from kimchi to yogurt and kefir, long prized for their preservative properties. Yet an underappreciated part of their appeal might be they harbor beneficial bacteria that aid digestion and the nutrient absorption. I think that's very well known and appreciated, but okay. The findings suggest that our appetite for fermented foods may stem in part from an ancient biological logic. When nutrients are scarce, the body appears to tune its sensory system to locate what it needs, sometimes by detecting microbes rather than macronutrients. Wow. It switched the target. Wow. From a living thing to a non-living thing or vice versa. Wow. Sensing flux, exactly. This study touches on a broader question in biology, how internal physiological states shape perception and behavior. The idea that hunger can alter sensory processing and neural activity isn't new, but this research goes further, showing that across multiple nutritional deprivations, certain sensory receptors are reprogrammed at the molecular level to enhance fitness. In other words, the fly's brains are not just interpreting hunger, they are physically changing the way they sense the world to meet it. Although we often think of these senses as fixed, they are in fact remarkably dynamic. Okay, I love these last two paragraphs. I understood a lot. So I'm gonna leave it there because almost done. And next time we're gonna check this, how the brain talks to the immune system. And dissociation is not only a symptom, it can act as an adaptive emergency brake when threat overwhelms. In PTSD, about 1 in 5 meet a dissociative specifier. Okay, I don't know what this specifier is. The personalization, derealization. Tough estimates vary by method. Okay, I'm gonna review that, and if I can understand it a little bit, maybe we can read it. We have the link of the original. It's a thread. Okay. Next time we try to understand at least some pieces of this thread. Sounds interesting or looks interesting. Thank you very much for your attention. It was a pleasure to me to be here again live, trying to improve my English. The 2nd of December, I have a new invitation to Quite Frankly, and we're gonna talk about CIA's The Alice in Wonderland techniques. Wonderworld, that's a song of Oasis, if I'm not wrong. We're gonna talk hypnosis and CIA's interrogation techniques in this era, using it now in social media to mass psychology endings. So I'm here again trying to improve my English, live reading and talking to you. Thank you very much. See you next time. Big hug.
Come on and cry me a river, cry me a river. I cried a river